So hi, welcome back to the Obscure Scholar YouTube channel. Uh, so we'll be continuing with our second video of biotechnology. So just a disclaimer: this is not a typical, a tutorial video. I am doing it for my own revision purposes. So yeah, let's continue with human reproduction. That is the second class of biotechnology. So as we've read in the previous class that a cell, in order to call something a cell, it should have three basic criterias. That is, it should have a cell membrane, it should have a cytoplasm, and it should have genetic material, right? And genetic material, stores the information to run a cell, right? And uh, every cell contains DNA, and DNA is condensed to form chromatin. And during cell division, only during cell division, the chromatin forms chromosome by replication, right? So we'll deal with chromosome in further as well. So yeah, so what happens during cell division? So suppose we have a cell and it has x amount of DNA in it, right? So or x amount of chromosomes in it, right? So during cell division, so the logical answer should be if a cell is getting divided into two, right? So each of the divided cell should have x by 2 amount of chromosomes, right? But that doesn't usually happen. So as we have as we have studied, each cell uh, is capable of functioning independently as well, right? So if it has x by 2 amount of chromosome, of course it won't be able to function independently, right? So after cell division also, x chromosome, the daughter cell will have x number of chromosomes only. That is the thing which is written over here. The daughter cells will have identical genetic complement. The daughter cell will also need x amount of chromosome to function independently, right? So coming up, so humans are made of cells, or everything is made of cells only. And most cells have DNA. This is, we have just read, DNA will convert to chromatin, not convert. DNA coils, as we have read in the previous class. It coils to form chromatin around a histone protein, and from there further coagulation gives rise to chromosome. So yeah, so what is the distinct characteristics of species? So the number of chromosomes a species has gives the species a certain character. For example, a roundworm has only 4 chromosomes, right? Whereas rice will have 24 chromosomes. Humans will have 46, and chimpanzees will have 48. So that is the reason, humans and chimpanzees are so similar. Not similar, but they are identical, right? Not exactly identical, but you can understand what I mean, right? So see, the difference is very less. So yeah, so each cell is made up of, as we have read, human cell will have 46 chromosomes in it, right? So each cell is made up of 46 chromosomes, 23 from the father, or from the female, and 23 from the male, right? So this 46 chromosomes can also be written as 2 into 23, right? Or 23 pairs, or 2n. So this 2n configuration of DNA is also known as diploid, right? So we have 46 chromosomes, and from there we have 23 pairs, that is 2n, that is diploid configuration. So what actually happens during cell division? So there are two different processes for cell division. One is mitosis, other one is meiosis. So during mitosis, what happens? So here we have a cell, right? We have 2 into 23 chromosomes. So during cell division, the chromosome first doubles itself, right? So here, suppose this is a cell. this is a cell. What happens is, if I draw the diagram, first the chromosome replicates itself. It will do like this, right? So this is a cell, right? And first it has replicated itself from here the chromosomes will split itself, right? So from here we'll have a structure like this, a chromosome like this, and from here we'll have another chromosome like this, and two different cells are getting formed, right? So this is what happens during mitosis. So yeah, during cell division, we have seen, so from here, first it will double itself or replicate itself. So from 23 it will become 92, and then further during cell division it will again come back to 23, that is 46 chromosomes, right? So this entire process is called mitosis. Mitosis happens during cell growth, right? So we have seen from 2n to 4n, and then finally it gets divided into 2n to n, right? So let's see what happens during reproduction if we use the same method only. If same meiosis is getting used, so what happens? So here we have the sperm cell, we have the egg cell, both getting fused to form the first cell, that is zygote, right? So as we have seen in meiosis, what happens? So sperm cell will have 2n configuration of DNA. Egg will also have 2n. Both will fuse. So when both will fuse under meiosis, it will become 4n. The zygote will have 4n, and thus it will have 92 chromosomes. So of course that is a problem, right? Because humans should have only 46 chromosomes, right? So that is the reason mitosis does not happen during cell division. During cell division, what we see generally see is meiosis. Not generally, every time what we see is meiosis. So in meiosis, what happens? So in mitosis, we have seen a cell gives rise to diploid configuration of cells, right? So from 2n we have 2n to n, right? But under meiosis, what we'll have is first here we have 2n configuration. So first it will replicate itself, the chromosomes will replicate itself, to 4n, and then from 4n 4n cells will get formed, and these are called haploid configuration. These cells will have n number of chromosomes, and thus when the n gamete will fuse with the n gamete, that is the sperm, and n gamete of the female, that is the egg, when they fuse, it will form a zygote, that is 2n, right? Again from here, if we see again, the zygote will again grow under mitosis, and it will again do cell division. So again then we'll have mitosis and not meiosis. Meiosis will only happen when we'll have reproduction, right? So that is simply what is written over here. Cell will it will make two haploid cells in meiosis during cell growth, but during reproduction meiosis will take place, four haploid cells will get generated. So yeah. Okay, so now the obvious question. So every time the father is giving half its DNA, and the mother is giving half its DNA, and we have the child cell, right? So when we have the child, the genetic material will have the information about the height of the child, the skin complexion, and everything else, right? So whenever the parents are deciding to go for their second child, the same half DNA is going there, only half DNA of the father and half DNA from the mother, right? And the cell is getting formed, the cell of the child is getting formed. So thus both the child should look identical, right? Should look exactly the same, right? But that doesn't usually happen, what we have seen. So that is because of the concept of crossing over. the child does not look exactly the same. That is because of crossing over. So what happens is, every time during sperm formation, the DNA undergoes a little bit of change, right? The DNA changes just a little bit, and this entire process of the change in DNA is known as DNA crossover. So that is the offspring, the first offspring and the second offspring. They don't look exactly like the same, but they are very similar, because the DNA change which is happening due to that crossover is very very small, right? So yeah. Okay, so here we have some basic information. So the time taken for cell division is 90 minutes in case of yeast, and the cell division for human cell in culture means in laboratory, right? In lab. So we have so it is 24 hours, and some cells do not divide. That is the heart cells; they do not divide. Coming to the next topic, so here we have a sperm cell with N chromosome. Here we have the egg. Okay, egg has N chromosome. It has mitochondria. It has endoplasmic reticulum. It has Golgi body, right? So this is the sperm. This is the egg. So during the fusion, the reaction that happens is acrosomal reaction. So now many DNAs come, and only one is allowed to enter, and that the reaction that facilitates that fusion is called acrosomal reaction. And one thing we must understand, that is the cell membrane does not allow the entire sperm to enter the egg, right? It only allows the nucleus of that sperm to enter the egg, right? So here, when the nucleus of the sperm enters, so here we have the new egg, that is the zygote, with n configuration. So here we have 2n configuration, right? So this is the zygote here. So a zygote will have two type of DNA, right? It'll have nuclear DNA and it'll have mitochondrial DNA. So nuclear DNA, the zygote will have from both the mother and the father. So you have seen here, this is from the sperm from the father, and this is from the mother, right? But the mitochondrial DNA will be exclusively from the mother. So now the obvious question is, since the offspring or the zygote has, more of mother's DNA, so the zygote or the child must look more like the mother, right? But that does not usually happen, right? So that is because the mitochondrial DNA or the mitochondrial gene that is there, that is very less. We have 20K genes in total, but only 37 among them is mitochondrial DNA, right? So yeah. So now another question: what if during cell division or during reproduction, the mitochondrial DNA or the mitochondria is faulty, right? The mitochondria is defective. So the obvious solution is mitochondrial replacement therapy. So in case the mitochondria is faulty, what happens is, during that faulty mitochondria only divides further, right? So that gives birth to Lay's disease, right? So what is the solution? It is mitochondrial replacement therapy. So there are two basic systems. Okay, so here we have the sperm cell. Just a second. Okay, so here we have the sperm cell, and here we have the egg cell, right? In mitochondrial replacement therapy, what happens is, uh, first the cells are allowed to fuse, right? Okay, so the sperm and the egg will fuse. Here we have the nucleus of the sperm, here we have the nucleus of the egg. First they are allowed to fuse, and here a pronucleus is formed, right? A pronucleus is not a fully developed nucleus, right? It is just before the nucleus is formed. So now here on what happens is we'll have to identify a donor's egg, right? We'll have to identify a donor's egg with a good mitochondria, right? Then this pronucleus should be replaced by the donor's egg's nucleus, right? So this nucleus will be replaced by this nucleus, right? So now the new zygote that is being formed, it has the right ribosome, the right mitochondria, the correct mitochondria or the good mitochondria, and it has the mother's and the father's nucleus, right? The 2n configuration nucleus, right? So this is how new zygote is being formed, and this entire process only is called IVF, which we did and which we listen. The full form is in vitro fertilization, right? So this is also called pro-nuclear transfer. Pro-nuclear because here the nucleus, we are transferring before the nucleus is being formed. It is also known as three parent baby, right? So there is another solution as well, okay, that is called reconstructed egg technology. Here what happens is, so here we have the sperm cell, here we have the egg cell. So in the previous one we have seen that initially in the solution one, what happened was we had allowed the egg and the sperm to fuse, okay, and then the pronucleus was getting transferred. But here we won't allow this fusion, right? This sperm cell will would not be allowed to fuse with that egg cell. So here again a donor egg is identified, and right at the beginning only the this nucleus of the female is being replaced by the nucleus of the donor's nucleus, right? So from here we have this. So here we have the sperm cell, here we have the donor's egg with the mother's nucleus, right? So here this is how zygote is getting formed. So this is also known as the three parent baby. So this is the two ways through which IVF is done, right? So there is another process as well, another problem as well. So what we have seen is uh, so this is the vagina, right? This part is the vagina, this is the cervix, this area is known as the uterus, and this is where the fusion happens. And here, from here, the egg starts to go through the fallopian tube. So what happens when this fallopian tube is blocked? For some odd reason, the fallopian tube is getting blocked. So what will happen is the egg won't be able to transfer it and get fused here with the sperm, right? So here also the same thing is done. The egg is taken out, the sperm is taken out, okay. The zygote is being formed through IVF, and that zygote will be placed over here, okay. So this is also another way through which IVF is used, okay. So then we'll see two different questions, PYQs. So here we have in the context of hereditary disease, consider the following statements. Passing on mitochondrial disease from parent to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization of the egg. A child inherits mitochondrial disease entirely from the mother and not from the father. So both 1 and 2 is correct, right? So in context of recent advances in human reproductive technology, pro-nuclear transfer is used in. So this is prevention of a mitochondrial disease in offspring. So now let's come to understanding genome and genes.